Monohybrid cross. It is a cross between two parents for one character and its two traits. The cross between two parents for one character and two traits. This is monohybrid cross. Let us take parental generation. Capital T for tall plant and small t for dwarf. In F1 generation, we will get all the capital T and small t. Either we can directly cross it like this. First with first, first with second, second with first, and then second with second. Or you can directly make Punnett square. So in this case also you are going to get the same result. All the F1 individuals are tall, and this is because of law of dominance. Because one dominant allele masks the effect of recessive and the dominant is only going to express. Now, in order to get F2 generation, we have to perform selfing. Now, what is selfing? You have to take two members of F1 generation and allow them to cross. So, by this way, or again by making Punnett square, you will be getting the recombinations in this way. So 1 ratio for homozygous dominant or homozygous tall, 2 ratio for heterozygous dominant or heterozygous tall, and then again 1 ratio for homozygous recessive. This is how we get F2 generation. So this is the genotypic ratio. This is a genotypic ratio of monohybrid cross. But when we talk about phenotypic ratio, then phenotypic ratio is 3:1, because either in homozygous dominant or in heterozygous dominant we are getting three individuals tall and one is dwarf. So 3 will be tall and 1 will be dwarf. This is the phenotypic ratio and this is genotypic ratio. Now, in order to get the phenotypes of monohybrid cross, there is one formula also, that is 2 raised to the power n, where n is the type of cross, and for monohybrid cross the value of n is always 1. So like this we will be getting two phenotypes. One phenotype is tall and another phenotype is dwarf. So this is the formula for the monohybrid cross in order to get the phenotype. Now if we want to get the genotypes, there will be little bit change in the formula, that is 3 raised to the power n, where again n will be 1 for monohybrid cross. So 3 genotypes are there, as you have seen: capital T, capital T, capital T, small t, and small t. So in order to get phenotype and genotype, there is also these formulas. Similarly, there is one formula in order to get the offsprings also. So in order to get the offsprings, the formula is 2n whole square 2 1 by 2. Then in this case, total you will be getting 4 offsprings, which is clear here. The number of offsprings are 4. So why I am telling you these formulas is only from the NEET point of view or from the objective point of view. If you get the question in objective, this is easy way, right? So thank you. My next video will be of dihybrid cross. Dihybrid cross. Dear students, in dihybrid cross, we have to take care of the word two. Di means two. So here we are making a cross between two parents for 4 traits of two characters. There will be 4 traits of two character for which we perform a dihybrid cross. So let us take a parental generation, capital R, capital R, capital Y, capital Y. This is round and yellow. We will cross it with wrinkled and green seeds. This is parental generation. Now the F1 generation is always a mixture of parental generation, and in all the member of F1 generation we are going to get this type of recombination of alleles. So all the F1 generation will be round and yellow because of the law of dominance here. Now students, in this case of dihybrid cross, the next step is you have to make gametes. So the gametes would be capital R, capital Y, capital R, small r, capital Y, and at last small r, small y. After this, we have to make Punnett square having 16 blocks, and we have to write the genotypes outside. Now taking one genotype, we will cross it with the rest and put the sequence here one by one. So like this, we have to take out different genetic recombinations, and we will get total 16 offsprings in it. These 16 offsprings will be having different genotypes. Now when it comes to F2 generation, then we have to count that what type of individuals will be round and yellow. So the one which are having single R or double R will be round and yellow, and the dominant one. So 1 2 3 4 5 6 7 8 9. So here 9 we will be having round and yellow. Then after that, round and green. Let us take these recombination with small y. So this will also be round but green, and this will also be round and green, and here the ratio of round and green is 3. Then next, let us see wrinkled and yellow. So let me put a star mark here. So this is wrinkled and yellow with capital Y. Here also wrinkled and yellow and wrinkled and yellow. So 3 we are having wrinkled and yellow, and at last the one, this one, the last one, this is wrinkled and green. So 9 is to 3 is to 3 is to 1 ratio is purely because of the law of independent assortment. But now again, from objective point of view, if you come across the question that you have to find the number of phenotypes produced in a dihybrid cross, then for phenotypes the formula will be same, 2 raised to the power n. Here because it's a dihybrid cross, so n will be 2. So you will be getting 4 phenotypes in this case, that is round and yellow, round and green, wrinkled and yellow, and wrinkled and green. These are the 4 phenotypes, and this is also known as phenotypic ratio. 9 is to 3 is to 3 is to 1. If you have to find out the genotypes, then it is 3 raised to the power n. So that is n will be 2 for dihybrid cross. So 3 into 3, this will be 9. So there are 9 genotypes in a dihybrid cross, right? And the ratio is not in syllabus, but let me tell you, it is 1 ratio 2 ratio 1 ratio 2 ratio 4 ratio 2 ratio 1 ratio 2 ratio 4, right? And at last, if we talk about number of offsprings, then so in order to get the number of offsprings in dihybrid cross, we have a formula, 2 raised to the power n whole square. So this will be 4 whole square, and total we will be getting 16 offsprings. So in order to get offsprings number in dihybrid cross, we have this formula.